what we found is that in the absence of one of these key genes that is missing in ALS, that two of these genetic codes basically interact aberrantly or in a wrong fashion with each other. So the DNA essentially gets stuck to the RNA, and this in turn creates a lot of problems in the cell and obstacles to other things. In these healthy cells, what is happening is that this RNA is able to float away from the DNA because ataxin 2 binds to it and makes sure that it takes it away from the DNA. However, if you look now on the other side of the image in the ALS cells, the RNA, as you can see here, is generated by this, again, the same enzyme, the big blue enzyme, but it gets gummed up and stuck on the DNA itself, and this basically create these obstacles along different parts of our DNA, and these are highly destabilizing to our entire genetic code, and that's what is leading to these problems in cells that are lacking this ALS gene called ataxin 2. These increased hybrids that we see in these ALS type cells feedback and cause all sorts of problems in the cell with the RNA, and then basically it's a gumming up of the RNA. We were able to discover that if we decrease the calories that we're giving cells, ALS type cells, to the minimum necessary amount, we're actually able to disrupt that race and basically disrupt this driver of ALS. So going forward, we're going to be looking forward to developing more targeted therapeutics to disrupt this race at various steps. You need different runners to be constantly passing on the baton, say, to each other in a race. So technically, if you can just get one of these runners to drop the baton, the race is done. And that's really the goal, is that we get someone to drop the baton, and then they're automatically disqualified, and we hope we can disqualify ALS.
Our finding is very basic in terms of, we find a function for this ataxin 2 protein, but that concept can be extrapolated to many other proteins and many other diseases, not just ALS, even Alzheimer's disease, even Parkinson's disease, Huntington's disease. They have similar processes where people didn't actually know or still don't know what's going on. But this mechanism that we propose in our paper could be the underlying mechanism for a wide variety of neurodegenerative diseases.
The next steps are already happening in my lab. So we are looking at the applications of these findings to cells and tissues from ALS patients. And I can say preliminary results are very encouraging. On another front, we're developing various screens so that we can try to mimic the reduced calorie effect that we saw in ALS cells and try to develop more robust therapeutic approaches.